Okay, thank you very much. It's really nice being here after 4 years again. So my talk is going to be on ultrasonic vocalization in mouse models for autism spectrum disorder. And the common thing in the models that I will present is has a focus on the synaptic dynamics. Since it is these are models for autism spectrum disorder, I will just briefly introduce. As you know, autism is a neurodevelopmental disorders, and major deficits seen are in the social communication and interaction, together with repetitive patterns of behavior or interest. This is the DSM-5 definition of autism spectrum disorder. And currently it is known to affect about 1 to 1.5% of individuals worldwide. And it is also known that there's a strong bias in males. The current estimation is that the 40 to 80% of the cases have a genetic cause. This is a large span, mainly because it's quite a heterogeneous disorder, and in some cases the mutations are actually not very clear. Therefore, the 40 to 80 wide range. But as you can see, these are mainly the factors that contribute to this genetic susceptibility in humans. So it can be epigenetic modifications, it can be double hit mutations or copy number variations, CMEs. There can be environmental factors which in turn also affect genetic mechanisms. This can be de novo mutations. And there's, of course, also sex-linked effects, like some hormonal effects and so on. And here is a list of a few genes. But actually, if you browse through the Safari genome, human gene database, I don't know if you can see clearly, but there's around 1,100 genes which have been definitely linked to autism spectrum disorder cases. And what is also known is that most of those genes actually converge in pathways which are related to transcriptional or translational regulation. They are involved in signaling pathways, in actin dynamics, and synapse homeostasis. So some of the genes which was shown in a paper by Elodie is showing how they actually contribute in the synaptic pre or postsynaptic domains. And what is listed here are the mouse models that have been generated for these genes. So one of the genes in terms of the synaptic communication is that I'm going to focus are the family of Schenck genes, which encode the Schenck proteins. These are postsynaptic scaffolding proteins, which mainly, as you see here, hold the synapse together. And they are through connecting via Homer. They are connected to such as glutamatergic receptors, but they are also involved in regulating the actin dynamics in turn. So they are also connected, of course, to the NMDA receptors at the membrane or the neuroligands. So they basically hold the synapse together. And in humans, also several point mutations in Schenck genes. So there's a family of 3 genes, Schenck 1, 2, and 3, and they were all associated with autism spectrum disorder. And again, in the same database, if you look at the scores for how much these genes are involved, you get a gene score. So as you can see, the Schenck 3 has actually the highest score, so it has been associated with many cases, whereas Schenck 2 is a bit less, and Schenck 1 is a bit less too. Nonetheless, we have looked at these mice too, but I will explain that later. So I've shown you that the Schenck genes are connected to the actin F-actin, so the actin filaments that hold the synapse together. And there are also two major proteins at this domain, which are the actin binding proteins. These are cophilin and profilin, which are involved in assembling or disassembling of the F-actin. So F-actin is the active form of the actin filament, so from G-actin to F-actin, which needs to be tightly regulated by these two proteins because you don't want too much of actin filaments forming. So in that case, cophilin comes into place, or when new filaments are forming, profilin takes into place and builds this tightly hold synapse. Cophilin and profilin are not the only actin regulating proteins, of course, but there are some other actin regulatory proteins, and these proteins are also shown to be associated with autism spectrum disorder, which makes cophilin and profilin proteins as well of interest to look at. And then there are the microRNAs, which are one of the key regulatory processes in post-transcriptional regulation of certain genes. So microRNAs, these are small non-coding RNAs, which, as I just said, they regulate the post-transcriptional gene expression, and mainly they are, as you can see, they are the pre-form of microRNAs. They are exported from the nucleus into cytosol, and then by binding through the silencing complex, they can actually either lead to mRNA cleavage, so a regulation of the mRNA transcript, or they can actually also suppress the translation completely. In the microRNA world, so there are also hundreds of microRNAs, which all have specific roles, but there's a specific microRNA cluster which encodes for 38 different microRNAs, and within this cluster, some of them were also associated with neuropsychiatric disorders. So this is why this big cluster is quite interesting, because also the expression of the cluster was shown to be regulated by synaptic activity. So this is why they are also majorly involved in holding the synaptic activity in place. We work with those mouse models, which are relevant to the three major domains that I just showed you: Schenck, cophilin, profilin, and the microRNAs. And in order to test their behavior, whether it is linked to any autism-like behavior, we look at certain domains which are implicated in humans. So these are the communication impairments in social behavior and the repetitive behavior. But for the sake of this conference, I will focus on the two domains, which is communication and social behavior, and how the two are actually linked. I mean, you have already heard the different type of vocalizations. Since we mostly look at the infant mice and juvenile mice, I will only talk about this, and you already heard from Sylvie's talk about the adult vocalizations. The infant, the pup vocalizations and the juvenile vocalizations mainly differ in the frequency range, but also in terms of the let's say the shape, so how much they span the entire spectrogram in duration and in the also in the frequency. So the pup vocalizations, the most common way to test that is the isolation from the mother and the litter mates, whereas the juvenile vocalizations can be nicely tested in terms of social interaction, as you have seen from Sylvie's talk as well. We have the pup vocalizations we have investigated in various mouse models. The first one was the Schenck-1 knockout mice. where the pups were, in this experiment, isolated from the mother and the litter maze and tested on a clean bedding for 10 minutes. And what we have seen is that the knockout mice, depicted by the minus here in white bars, emitted in general fewer vocalizations in comparison to their heterozygous and wild-type litter mates. And we have tested these mice throughout several postnatal days, and in all postnatal days, we have actually seen reduced vocalizations, but especially on postnatal day 6, we had the strongest effect. After that, we wanted to look at the effects of social context, whether this actually plays any role in emitting the vocalizations. Therefore, we picked postnatal day 9, where we haven't seen significant differences, and even they were comparable levels of wild-type mice and knockout mice, and we tested the mice under different social contexts. So again, under clean bedding conditions, we haven't seen any significant differences, whereas if the mice were tested on a bedding, which was soiled with bedding from the home cage or from an unknown male cage, the pups had reduced ultrasonic vocalizations, which seems like the social context in this matter plays a major role in revealing those deficits. In this example, Schenk-1 knockout mice, we have seen reduced vocalizations, but actually when we look at the microRNA cluster knockout mice that I have just mentioned, we have seen an opposite picture of communication deficits, and actually the knockout mice, they were emitting much higher vocalizations across all postnatal days, and this actually was a consistent finding in a way, because later when we tested the same mice for at the juvenile age for their reciprocal social interaction, again we have found increased number of ultrasonic vocalizations, and they were actually also more social than the wild-type mice. We have also checked the juvenile reciprocal social interaction in the coffin knockout mice. So the protein that I mentioned, which is involved in disassembly of the actin filaments. Here, actually, we haven't seen any differences of both the control mice, but also the knockout mice, and as you can see also throughout the testing. So we actually apply a similar protocol. We isolate the mice, and then but only for 24 hours, as I mentioned. And then we test them for 5 minutes, and you see per minute, there's actually quite a lot of interaction going on. And this is also reflected in the number of ultrasonic vocalizations. And there's positive correlation between the behavior and the ultrasonic vocalizations. We looked at this in detail, both in wild type and knockout mice. So in this histogram above, you can see in black, you see the ultrasonic vocalizations emitted, and in red are the active social behaviors. So these are behaviors like sniffing, following, grooming, and so on. And there are some other non-social behaviors such as rearing, digging, or self-grooming. And both in wild type and knockout mice, we have seen that the social behaviors are reflected by means of the ultrasonic vocalization, in the sense that most of the vocalizations, so around 90% of the vocalizations, happen through an active social behavior. The same paradigm we have also applied in the profile in knockout mice, and here we have seen a completely opposite picture, actually. So as you see in this example, the conditional knockout mice, they had completely reduced ultrasonic vocalizations and also not so much social behavior. But interestingly, this was only the case in the typically social females. So in the female knockouts, we have seen the most reduction in terms of the ultrasonic vocalizations. The active social behavior was, in fact, also reduced, but this wasn't significant. yeah, so this was actually the three models that I wanted to talk about. Yeah, as I hope I was able to show you that these are actually three different models which are somehow linked together, actually, because microRNA cluster, microRNAs also regulate Schenk expression. So there's increased Schenk1 and Schenk3 expression as well in the cluster knockout animals. And Schenks are connected to covalin and profilin. So they not only converge in the synaptic role, so to say, but also converge in the behavioral phenotype when these genes are missing. And what has been very nice to see, I mean, there are now much nicer tools, I know that, but back then this was quite a tedious work, linking the USV and the social behavior. Yeah, this offers indeed a very nice tool to look at the communication domain in order to have a translational link between mice and humans. And yeah, I'm really looking forward to the next talks, be able to see to assess the ultrasonic vocalizations under those live mouse tracker and so on, how these are actually linked to social behavior of mice, and in order to have an even better understanding of the autism phenotype. Yeah, with that, I would like to thank a bunch of people. Most of the work I presented, I had actually performed back when I was in Marburg. So this was the group in Marburg, but three of us moved together with Marcus to Leuven. So now I continue with some other autism models, which I hope to present at some point again to you. Yeah, thank you for your attention. Jeffrey has a question, please.
Thank you. That was excellent. I have a question, but first I have a comment. You know, having done some of these experiments in Schenk animals doing autism and then linking that up with clinical trials, the clinicians really need our help. Right now, they just ask the parents, "What do you think your kids have?" And this is really for clinical trials. Just basically these remarks. So these studies will inform future clinical studies. Will be translation. And one question, what I've also noticed with some of these autistic mice, like the Schenk mice, is that they will vocalize more when they're hearing. But if I look at them in detail, it looks like it's abnormal social. So a male-female mouse interaction, if the male mouse is vocalizing more, but is attempting to mate, which is inappropriate. And I'm wondering if you've seen some of those things that you would be in vocalization with inappropriate.
Inappropriate behavior. I mean, actually, we haven't seen. So this was what was very striking to see from Sylvie's results. We haven't seen any sex differences in juvenile mice. Like they were always social. With Schenk mice, yeah. This was also the case, but also with the covalin mice, this was the case. So, yeah, the only difference we have seen was in the ultrasonic vocalization domain, but inappropriate behavior, I haven't, no, I haven't seen that, no. That's it.
Well, thank you for this talk. It's very interesting, and I just measure as well as you with their enormous amount of work that it is to for it, and we have the same thing that vocalization related to contact mostly. So it's I was wondering which background strain we'll be using because we know that there are many differences, and as you mentioned, I was also interested in understanding what would be the difference between our studies, so that there is no difference between the males and the females. But if there can be many differences,
yeah. So most of them have B6 background, actually, except for the Schenk mice. They have a mixed background, B6 and 129. Apparently these are not so good in vocalizing, you mentioned. But no, I mean, I would say they follow the usual pattern, normally with B6 background. So.
Testing them, because it was a question that people asked me, and I think it's a very interesting question. To test them in the classical cycle or the growth cycle, or is there activity period or?
No, we test in the classical cycle. I mean, because I don't know, when they are too active, sometimes, let's say at night, this is already something I also realize when I'm testing towards the evening or so, it already sometimes the mice get a bit hyperactive. But I mean, nonetheless, we try to dim the light, so we test under red light conditions and so on, so mimic a little in their active phase.
I have questions.
I can see up.
I think I saw some repeated patterns of vocalization in mice, multiple of autism disorder. Have you seen repeated syllable sounds that might not look as normal? I don't know if you see that.
We haven't done any classification, of course. Actually, I really would like to do that because I have a tremendous amount of vocalizations, especially from Schenk 1 occult mice. And I know that there is now the nice database to upload this USB webpage you just upload, and you can get the different clusters. Yeah, I would like to look at that. I just haven't had the chance, but there might be. Yeah.